So what I should do though, first of all, I know we're kind of very probably at the end to do a little wrap up, but want to thank, since I have the opportunity here at the podium, just to thank all the support that we've had in putting this symposium together, to thank our funders, Cancer Society and Science Foundation Ireland and industry, who've given us really great support to make all of this happen. But I'd particularly like to thank Sandra for the Trojan work that she's done in making this symposium work and be the success that it is. And I just wanted to say that now in case I forgot at a later stage. Kind of, it's been a long couple of days and probably had a later night last night than I should have had. So, but I won't say who's to blame for that. But I take personal responsibility. So in any case, I showed you a couple of these slides earlier. The initial sort of idea, I guess, was to try and develop collaborative research across the area of blood cancers in terms of putting early phase studies available for patients, having a biobank and having a registry. And since my real focus is the clinical trial side of it, this is what I'm going to cover in the next 10 minutes or 15 minutes or so. So we had the initial 3 sites, and then we were very fortunate that the Cancer Society made additional funding available to bring another couple of associate sites on board. So we now have a total of 5 clinical sites, and 3 in Dublin and 2 in Galway. But this was, when we had our initial opening back in November with the Minister, Mark Ferguson from SFI and Rob O'Connor from the Cancer Society, and I can name him because he was very no problem going public or anything. This is Paul Daly, a patient of mine with CLL, who has been through a number of clinical trials, and actually since this picture was taken, actually went on to have a transplant and has done very well actually from the transplant. But he recognised the importance of clinical trials and wanted to be associated, very happy to be associated and get the message out to patients. So I think it's important in terms of when we're looking at putting clinical trials available for patients in this space, seeing what the gaps are and recognising what's already there. And through Cancer Trials Ireland, I was about to say ICORG, a very good infrastructure already exists, and we're not trying to reinvent the wheel. The intention was always to complement what ICORG or CTI had in place. And so there are actually now quite a good portfolio of clinical trials available for patients with blood cancers in Ireland. And these are the studies that either have just closed or currently open or about to open in the multiple myeloma and AML space. And I pick out these 2 diseases because these were picked out as being where we were going to initially focus before broadening out into other cancers, into other blood cancers. But this gives you a flavour for what is currently there. So we are getting access to exciting, promising new drugs such as venetoclax, and the last trial that's mentioned there is actually one of these breakthrough designation studies, so recognised by the FDA as being a very important trial that's really meeting an unmet need in elderly AML, and you saw the data earlier from Alan that speaks to that. But in terms of what are the gaps, and this is the sort of thing that we hope to address with the network, is providing trials that do fill rather the unmet need. And so we've recognised that in fact in the late sort of refractory myeloma space that we don't actually have anything at the moment. We hadn't anything in relapsed refractory AML through the regular channels with Cancer Trials Ireland. And since the study has just closed recently in the transplant-eligible population, there's now a need for a study in that. So I'm going to quickly go through some of the studies that we either have open or about to open. So the first study is a trial using an E-selectin inhibitor in AML, and the rationale for doing this is that cancer, or rather leukemic stem cells, appear to bind to E-selectin expressed on blood vessels within the bone marrow, and this renders them chemo-resistant. On the other hand, the normal stem cells, when they come in contact with E-selectin, actually go into cycle. So if you use an E-selectin inhibitor, you can actually mobilize out selectively the leukemic stem cells while protecting normal stem cells. And so this was the rationale for going into a trial using an E-selectin inhibitor. This was the design for the trial and initial dose finding, combining this, in fact using this as an adjunct. So on its own, this drug wouldn't be expected to have any activity. The FDA and the HPRA here were prepared to accept this concept of an adjunct. And of course, I should say that healthy volunteer studies with the single agent, with the GMI 1271, were done and shown to be very well tolerated and not to mobilize stem cells. No CD34 mobilization was seen in the peripheral blood. But basically, we've got to the stage now where we have a recommended phase 2 dose, and where it is actually about to go into an expansion phase where we'll be treating more patients in the relapsed refractory setting, and where we'll actually also be treating fitter elderly patients with AML. But what I'm actually very proud to say is that we were actually the first site worldwide to open this study and got the first 2 patients on the study, and it's now open at the Fred Hutchinson Cancer Center in Seattle, and the Dana-Farber, and a number of other centres in the US, as well as one site in Australia. But the data are actually very, very promising. So in a relapsed refractory AML patient population, you might expect maybe a 30% response rate typically. It's early days yet. I mean, in a sort of phase 1 study, you can't get too carried away, but given the patients that I know were on this study, very poor risk patients, this is a very promising initial experience, and this data is going to be presented shortly at the European Hematology Association meeting. But the hope would be that we can expand this study out to other sites within the blood cancer network. So the next study I'm going to talk about is a study that will provide daratumumab in addition to a standard induction regimen for multiple myeloma for initial treatment of younger newly diagnosed patients. So we heard a lot of excitement this morning from Henk about the use of daratumumab and how really it is going to revolutionize the treatment of multiple myeloma. And as I mentioned this morning, it is now actually approved in Europe for advanced refractory patients with multiple myeloma. Speaking in a way a little bit to some of what Donald was talking about earlier, that more of a drug is not always necessarily better, and this is cyclophosphamide, an old drug. This is a recent review article which makes the point that in fact when you use low doses of cyclophosphamide, this can actually have very beneficial immunologic effects that are completely lost when you use higher doses. So in this article from one of the AACR journals, Cancer Immunology Research, they're sort of making the point that this is basically a cheap checkpoint inhibitor, that you can totally reactivate your immune system by using a very low-cost drug that is available to everybody, pretty much. Now, what interested us particularly about cyclophosphamide is the fact that when given to treat tumours, that it seems to induce a secretory response from tumours, such as ALL, for example. This data was generated in ALL, where the cytokines, chemokines released from the tumour cells actually lead to macrophage activation that allows macrophages to enhance their ability to kill antibody-coated cells. So we thought this would be a very good way of enhancing the activity of daratumumab in multiple myeloma. And Aideen Ryan, who many of you know, who has a keen interest in cancer immunology, generated very nice data, and actually a Cancer Society fellow, which allowed us to undertake a major collaborative project with Janssen around exploring this concept in the lab. But we're moving into, hopefully later in the summer with this clinical trial, moving this concept into patients. This protocol, it's a big learning experience. The university is taking on the role of sponsor here on behalf of the Blood Cancer Network. But this has actually gone to ethics. It's also at the HPRA, and we're hoping that this will actually be open by August in a few months' time. And this is a important collaboration as well with Cancer Trials Ireland. So they have done very important work getting this study to where it's at, and will help us run this study on behalf of the network. So this shows you initial induction with 4 cycles of treatment. Then patients have an autologous stem cell transplant. They then go on to have further consolidation, and then maintenance treatment along the lines of what Henk showed you in the study, the randomized study that the HOVON and IFM group are doing. This is the study scheme here. We're going to be using dose finding really around the cyclophosphamide, and then at the higher dose level, we'll be exploring if we can give a 1.5 milligram dose of bortezomib. And again, this is just the study schema here with the induction, the transplant, consolidation, and maintenance. But what I wanted to point out here is that since we're now getting to a stage where we expect that we'll get a large proportion of our patients into a complete response, just assessing complete response is no longer really sufficient. And we're not quite at the point where MRD, or minimal residual disease, has been fully accepted as a surrogate endpoint, but there is clear data that patients who achieve MRD negativity have much better outcomes, particularly when it's sustained. So what we'll be doing is next generation sequencing for MRD, which allows you to detect disease at a level of 10 to the 1 and 10 to the minus 6. And we'll be doing that sequentially. We'll also be doing PET CT scans on patients who achieve a complete response, as well as in-depth prognostic characterization of patients, including FISH and gene expression profiling at the outset. So really quite a detailed, intensive study. It's only 18 patients, but this is a question that we can answer hopefully quite quickly, get it out there and get it published, and within a couple of years' time, and that would be, I think, an important milestone for the network. Standard sort of 3+3 design based on what Donald kind of showed you as being the typical phase 1 design. We don't need to do any fancy statistical analysis, I think, in this particular study. So I won't bore you with the details of the dose limiting toxicity and so on. But what I want to do now in just the last few slides is tell you a little bit about another study that we're actually going to open on the 20th of June, which is an example of bench to bedside work that's actually done here. So this is work that was initially done by Siobhan Glavy, a very bright national SPR academic fellow, who worked on looking at glycosylation and importance of different glycosylation genes in myeloma, and with Siobhan we identified this particular cell transferase, ST3-GAD6, as being very important in multiple myeloma, being an independent prognostic factor for survival. So this led us down the path of investigating, well, what does this gene do? It's actually critically involved in generation of selectin ligands, and we found that in particular E-selectin ligands. So we thought that E-selectin potentially could be a good target in multiple myeloma. I won't go through all of the data that supports that, but Alessandro Natoni, who's here, has done fantastic work bringing this study on, or this story on, and in collaboration with Glycomimetics, we were able to basically show that when you take myeloma cells and you sort them or generate a population of myeloma cells that's enriched for the expression of E-selectin ligands, and put these then into an animal model, that the disease is much more aggressive. So this is up here. So these are mice, the survival of mice that have been transplanted with, in the red, E-selectin ligand enriched myeloma cells, or the parental cells here. So these E-selectin ligand rich myeloma cells, or cells, behave much more aggressively. And what is shown at the bottom here is that if you take bortezomib and treat the mice that get the regular cells, it improves their survival compared to control. But if you take bortezomib and put it into mice that have been transplanted with these aggressive cells, then it doesn't work. But if you add, in green here is shown the combination of the E-selectin inhibitor, which on its own, the blue line, does nothing. When you add it to bortezomib, in the green line here, now you can rescue those mice. You can basically overcome the resistance. So that led to us formulating a clinical trial with Glycomimetics to see if in patients who were slow responders beyond first-line therapy, so we're on a bortezomib-based regimen, who've had no better than a partial response after 3 cycles of treatment, or who are having a slow biochemical progression on bortezomib therapy, if by simply adding in this adjunctive therapy that would hopefully unlock the resistance by freeing up cells from the tumor microenvironment, we could actually improve the response. So that's what we're doing. It's very, very early days yet. We have a long way to go, and we certainly need everybody's support to continue the journey. So thank you very much.